Okay, I think that we will go ahead and get started. Thank you everybody for joining us for our IBSS virtual seminar this week. I just want to remind you before we get started to please mute yourself and turn off your video, and that this is being recorded. So you always have the option to hop off now and then watch later on our YouTube channel. So it is my distinct pleasure to introduce Dr. Carly Kenkel. Dr. Kenkel is an assistant professor in the Department of Biological Sciences at the University of Southern California. And I have actually had the pleasure of knowing Carly since 2014, so about 6 years now, because we overlapped at the Australian Institute of Marine Science, which was really fun and a great pleasure. And I got introduced to how wonderful the science is that Carly does. She received her PhD in ecology, evolution, and behavior from the University of Texas at Austin, and was an NSF International Postdoctoral Fellow at the Australian Institute of Marine Science before starting her own lab at USC in 2017. Her work focuses largely on understanding variation in physical characteristics of organisms, so phenotypic variation, and then how ecology or organismal environment or environment interactions select for different phenotypes, and how those interactions then implicate evolutionary trajectory of populations or of species. So I could go on and on about Carly, but without further ado, she is here to talk to us today about genotype and environment effects on growth and survival of Acropora cervicornis in the Florida Keys. So take it away, Carly.
All right, share my screen. Can you see this now? I'm going to assume yes. Yes. Awesome. So yeah, thank you, Rebecca, for that lovely introduction, and to Morgan for the invite, and Alejandra for coordinating all the seminar stuff. And I'm really excited to talk to you today about some science that's sort of very much in progress. Let me see if I can advance my slides. So take everything with a grain of salt. It's not finalized, but in turn, I'd really love to benefit from your collective expertise. So don't be shy about jumping in with suggestions for improvement in the question and answer section. And thanks to Rebecca's introduction, I can kind of skip this slide since you already know what my main interests are, right? Connecting the environment with organismal phenotypes and genotypes. And I will say, like some of the other researchers at Cal Academy, because we're working on the ecological equivalent of canaries in the coal mine, we are very much interested in translating findings for the conservation and restoration of reef ecosystems. So I'm really interested in understanding the interactions between genotype and environment, because this knowledge is really central for many areas of biology. If we know how a phenotype or how traits are produced by genotypes or the environment, that gives us really incredible power to predict or alter phenotypic outcomes. So for genetically determined traits, we can, say, predict the response to selection, or know in advance that an individual might be at risk for developing a disease and intervene in time to reduce some of the negative impacts. Whereas if we know that a phenotype is largely environmentally determined, then we can change the environment to, say, improve crop yields or limit pollutants. But genotypes can also vary in their phenotypic response depending on the environment they're exposed to. And this produces something that we call genotype by environment interactions, or commonly abbreviated as this GxE, or G by E. And these graphs are just meant to illustrate how we kind of visualize the effects of genetics and environment, or their interaction on a particular trait value. So these hypotheticals consider three genotypes. You've got like the red genotype, the purple, and the blue genotype, or individuals. And you can have cases where the traits differ as a fixed effective genotype, regardless of the environment. So we have trait on the y-axis and then environment on the x. And in this first panel, you can see that the red genotype always has the highest trait value. It's the same trait value regardless of the environment. So it's a pure genetic effect. And that's like pure predictive power, right? If you know who the red genotype is, then you can say something about its performance in any given environment. You can also have cases where genotype and environment have additive effects on phenotype. So here the trait is plastic, so it can change depending on the environment that it goes into, but the red individual still has the highest trait value. And so you can still predict the relative performance of these different genotypes in any of the different environments. You just might not know the absolute trait value for any given case. But this capacity for plasticity can also vary depending on genotype. And this is where you get genotype by environment interactions, where you have changes among genotypes in the rank order or the relative magnitude of effects in different environments. So you can see here in this last panel, for example, the red genotype has the highest trait value in one environment, but then it actually has the lowest trait value in another environment. And you can also see cases where the slopes aren't parallel, right? Where the trait value is changing more in the purple genotype across environments than it is in the blue genotype. And this makes predictions more challenging as you're moving across environments. To take it into real life, we've known for decades that coral are really highly plastic. They can shift their morphology in response to light availability, nutrient levels, or wave energy. But fairly few studies have actually replicated genotype in a way that would allow us to really robustly quantify interactions between genotype and environment. And similarly, we don't really know whether these changes in morphology are adaptive, right? Whether they actually influence fitness or the ultimate survival or reproduction of a given genotype. And the example I'm showing you here is one of the better ones from John Bruner and Pete Edmonds, where they transplanted 10 Madrasis genotypes, or 10 unique individuals of Madrasis, among 4 different reefs in Jamaica. And they found that transplants shifted their morphology. So skinny branching individuals started growing more club-like when they were moved to higher wave energy sites. But they also found significant genotype by environment interactions for various aspects of correlate morphology, so kind of the shape of the little skeletal structures in the coral. And this is illustrated in some of these like multi-site G by E plots here on the bottom, where you can see different individuals are having different trait values across the 4 different sites. Now, genotype by environment effects aren't always bad or confounding per se. In some cases, these effects can also be really informative, say when you're trying to test for local adaptation, because in this case, we're actually looking for a population level signature of a genotype by environment interaction for fitness. We want to see that, right, say in this case, individuals from inshore environments have the highest fitness at inshore reef sites, whereas offshore individuals would have the highest fitness at offshore reef sites. And the work I want to tell you about today in Acropora cervicornis was actually born out of work that I had done during my dissertation, which focused on testing for local adaptation of corals between these inshore-offshore reef zones in the Florida Keys. Why was I working in the Florida Keys? Well, I had all of this excellent environmental data showing that there was a very big contrast between inshore and offshore reefs, where we knew that inshore reefs got hotter in the summer and colder in the winter just about every year. They also had higher levels of nutrients like dissolved in organic nitrogen and phosphorus, as well as higher levels of turbidity. So I came up with this brilliant design where I reciprocally transplanted corals, specifically the Coralliporides asturides, between inshore and offshore reefs. And I also thought I'd be clever and include these alongshore controls, so that I could show that it wasn't just a site-specific effect, right? It wasn't just an effect of the particular sites I had chosen, but it was this overarching pattern of local adaptation by the inshore-offshore reef zones. So I thought that inshore individuals would perform equally well at either of these sites, but then the big change would come when they were moved to offshore reefs and vice versa. I got my significant genotype by environment in effect. I saw that native individuals, so native transplants, had higher growth. In this case, this is weight gain in the inshore and offshore environments, respectively. And I was really excited. And then I plotted the alongshore controls and saw this pattern, which was really surprising because the corals actually hated the alongshore transplants more than the reciprocals. And I still don't have a great answer for this because all of the environmental contrasts, like almost all of the big water quality players, like nutrients, turbidity, salinity, dissolved oxygen, nearly all of the temperature data would say that the inshore-offshore contrast is the greatest in terms of like environmental stressors. And yet, both populations still showed these like much greater negative reactions being moved to another site within their same environmental zone. So in thinking more about this later, after I had graduated and published the original study, these population level averages mask some really interesting patterns at the individual level. So I replotted this population level data to now show you individual reaction norms. So inshore individuals are colored in red and offshore individuals in blue. And this is one of the really cool things about corals is they reproduce via fragmentation. So you can actually perfectly replicate genotype across environments. And so in this experiment, I only had a single replicate of each of my genetic individuals in each environment. So, I can't perfectly quantify the effects of genotype because I just haven't replicated it. But there are some fascinating patterns, right? You can see that there is an additive. There does seem to be an additive effect of genotype, right? Some individuals are performing better on average at all sites than others. But there's also individuals that are kind of bucking this population level pattern, where you have this offshore genet who actually really loved the alongshore transplant, counter to all of the other offshore individuals. And I wondered if similar dynamics could kind of explain the variable patterns of growth and survival that we see in transplants that are made for the purposes of reef restoration. And very briefly, why are reefs being restored? Well, the loss of coral in the Caribbean has really decimated both the structure and function of reef ecosystems. Historically, reefs in the Caribbean were really structurally complex, right? You can see examples here from Discovery Bay in Jamaica and Carey Strait Reef in Florida, where you have these branching acroporids, Acropora palmata and cervicornis, just covering reefs. But in the last 40 years, climate change, eutrophication, algal growth, storms, disease, hurricanes have resulted in significant declines. And in the Florida Keys today, less than 1% of surveyed corals are branching acroporids. And this changing reef structure has really negatively impacted the ecology. And that's because the complexity of reefs is correlated with the diversity of other reef organisms. For example, the abundance and richness of fish species is higher when you have a more complex reef structure. And this can influence the productivity of fisheries. And so really massive efforts are underway to try to restore these ecosystems and their services by planting corals back on reefs. So corals are mass propagated in both land and in water nurseries, and then the little fragments are transplanted back onto reefs. But those transplants don't always perform like you would expect. And so this graph on the right is just showing some pilot data that we got from a collaborator in the lower Florida Keys, where I had done my original work, showing that you can have wildly different survival outcomes. So, 83% mortality at one site versus only 3% mortality at another. And these sites aren't that far apart. And again, they're all within that same environmental zone. And about the same time that I was synthesizing all of this information in data, a study came out that was led by Ford Drury when he was at the University of Miami that found that there were no genotype by environment interactions in growth or survival of Acropora cervicornis in the northern Florida Keys. And so that suggested that, right, if you could collect some information on genotypes and their performance, you might still be able to predict restoration outcomes. It might just be a matter of which genotypes you were selecting. And so in January of 2017, before I started my lab here, I wrote a proposal in collaboration with Corey Credit at Eckerd College to ask what was driving differences in coral performance along shore in these lower key sites, and whether genotypes did in fact respond additively to variation across sites. And so we just wanted to quantify as much of the variation as we could. We aimed to look at growth and survival in replicate Acropora cervicornis genets following transplantation, and examine both abiotic, so like environmental water quality parameters, as well as biotic environments, so looking at microbial communities. And we also wanted to see if we could find some other predictive correlates. Specifically, we were looking at gene expression, but I won't have time to talk about that today. And before I get into the data, I want to acknowledge my amazing team that's processed all of this massive data set. So Emily Aguirre and Wyatt Million are both working on their PhDs in my lab, and Sibyl O'Donnell is an undergraduate. And Emily's been looking at genotype by environment effects on coral microbiomes for the first chapter of her dissertation. And so the microbial data I'll be presenting was all worked up by her. Wyatt is exploring the role of morphological plasticity and its mechanistic basis, and he did an incredible amount of methods development work to move this study beyond just changes in linear coral growth. And then Sibyl had started playing with the environmental data during a remote REU she did this summer through the Wrigley Institute at USC. And then finally, I want to acknowledge the continued support of Eric Bartels, who heads up field operations for Mote Marine Labs field station. We relied on his expertise to help pick the sites, like pick microhabitats, and Eric's just always gone above and beyond when it comes to research support. So if you are planning on going to the Keys, I highly recommend Mote, and specifically Eric, for helping coordinate your studies. So in April 2018, we began this field transplant, and we started with 3 replicates of each of 10 genotypes of Acropora cervicornis, and we transplanted those to 9 different sites in the lower Keys. And these are shown here in this map as colored circles. And the actual site names are listed next to it. The numeric circles correspond to fixed water quality monitoring stations. So we specifically chose these sites because they overlap with a long-term data set that is collected by the Southeast Environmental Research Center, where they come out quarterly and look at a whole suite of water quality parameters. And they've been doing that since 1995. So while it's not super high resolution in terms of being able to capture like particular pulse events, it is like the best environmental characterization that we have for the Florida Keys. And so that's why we chose these particular locations. At each site, we set up corals in these 3 replicate arrays. So like genotypes were kind of randomized and their position in each of these arrays at each site. And that's the replication design. We measured growth and survival at multiple time points. And the data that I'm going to be showing you today is through the first year of the transplantation. And that's when we saw the most change in survival thus far. And for cervicornis, these genotypes were originally wild collected, but they've been continually propagated in an in situ nursery. And that's the little triangle here with the N, for a minimum of 5 years, and in some cases up to 10 years prior to the start of this transplantation. And the study was originally funded for cervicornis, but I wanted to mention that we were also able to outplant 5 genotypes of Acropora palmata. And I'm particularly excited by the palmata because they're actually the result of sexual production. So these are first generation larvae from a Northern Keys population that were grown out and microfragmented in a land-based nursery since 2013. And our transplants, this is the first time that they've ever experienced a natural reef environment. So they've had like perfect environmental control. And I don't have any of those data today, unfortunately, because we got some funding later. We're still in the process of generating the growth models and sequencing data. But I'm hoping that the data will be ready in time for next summer's Coral Reef Conference. So that's just an advertisement to stay tuned. For growth, we actually measured growth using 3D photogrammetry because it allowed us to access more morphological traits than just linear extension. And for those of you that aren't familiar with this method, the concept's fairly simple. The idea is that you can recreate the three-dimensional structure of an object by taking still photographs from multiple different angles and then just including a size standard. So we're using Agisoft Metashape. There's other different softwares available, but this one has been particularly popular for like reef scale analysis. And we've just adopted it for individual corals. And what we do is we incorporate fixed positional targets. This is just a for example to provide a reference point for the individual coral. And for small ones, you know, somewhere between like 50 to 75 photos is sufficient to reconstruct a decent model. On a larger, more complex coral, we found like a few hundred photos is necessary, which sounds like a lot, but it doesn't actually take that much time. It's somewhere on the order of like 2 to 3 minutes or up to 5 minutes, depending on if it's a big coral. But we have detailed protocols available. If you're interested in applying this in your own work, you can just go to protocols.io and search for our method. And we've been using a high performance computing system to serially process the photo sets to create models so that, you know, the construction is kind of automated and happening in the background. And we then use the freeware mesh lab to measure focal traits. And this video just kind of illustrates the process and shows the level of detail that these models have in them. So we use the fixed references to scale the models so that it's actually to size. Then you just get rid of this background so that you only have the coral itself. And then you can take various trait measures. So the traits that we're measuring are total linear extension. And this is just the sum of all of the individual lengths of branches. And why we're doing this is typically the only method that you can take in the field. So you hang out with a pair of calipers or like a bendy ruler while diving and try to measure all the lengths. And so in our case, we can recreate those measures using a 3D model. So you can relate it back to like purely field-based data sets. But once the corals get really big, hanging out and measuring TLE by hand is really painful and in some cases impossible because there's just too many branches. And so having these models is a nice way to scale up. It's also possible to get surface area and volume from the same model. And then this last trait that was shown in the video is a more complex metric of like the habitat that a coral creates. So it's sometimes called the volume of the interstitial space. And the idea is it's almost like dropping a blanket over your coral and asking how much free space is left over apart from the coral itself. So sort of what would be the habitat for a fish or something that would be swimming about. Start playing the video again. And so as a for example, for this more complex branching morphology on the left here, there's going to be more of that interstitial space, where on the right, this coral is going to have less interstitial space. And to kind of foreshadow the punchline, these ramets are actually the exact same genotype that have been grown out for a year at 2 different sites. So the one on the left was at Big Pine and the one on the right was at Eastern Sambo. So you can see there's definitely plasticity in morphology for our sites. But getting into the survival results first, this is showing you mortality curves. So on the y-axis, we have the fraction surviving over time and, fraction surviving over time for the different sites. And there's definitely significant variation here, lots of variation across sites. The biggest decline was evident in the first 3 months. So when corals were the smallest, that's when we saw the biggest declines in mortality. And then the relative change has been smaller after that. We certainly have some best performing sites. So in this case, Eastern Sambo, for anybody that knows, is one of the better performing sites. And we had significantly different sites, so significantly worse performers in comparison to Eastern Sambo. And Bea Honda was one of the worst. And this wasn't hugely surprising because this was a similar pattern to what we'd seen in the original pilot data. where Bea Honda was one of the highest mortality sites. There's less variation among genotypes. So this is the same data, now breaking it down by genotype instead of site, and the numeric identifiers are just the different genotypes. And now they're colored based on their ranking. So bluer is higher surviving genotypes on average, and red is poorer surviving genotypes on average. And we do get some differentiation among genotypes, where we have our best performing genet 36 with about 90% survival across sites in comparison to 62, which is only like somewhat less than 60% survival across sites. There does also appear to be genotype by environment interactions for survival. So this is getting a little crazy, but now I'm breaking it down to where you can see survival for genotypes across the site. So I've retained the original coloring scheme where 36 is the top performer, like blue is the top performers, and then red is the poorer performers. And on average, but you can see across sites, you have instances where the poorest performers actually do just fine at our best site, Eastern Sambo, right? The darkest red is actually surviving perfectly well at Eastern Sambo. And other instances where it performs totally differently, right? Where you have, in some cases, like much higher mortality, and also instances where you have some of the top performers, right? Some of the dark blue genotypes not surviving as well. So there's certainly G by E for survival, and we're working on getting the most appropriate model for this that incorporates a time effect. We also see significant genotype by environment effects for growth and morphology. So as a for example, I'm just showing you the change in surface area. And these get a little crazy, these G by E plots, because I'm showing you lines connecting each individual genotype growth on average across the different sites. So again, normally we look at G by E plots for just 2 sites. This is kind of for all of them. To simplify, I'll just pull out 2 genotypes so you can kind of see some of these patterns. But what we found was that for any of the more higher order traits like surface area volume or the volume of the interstitial space, you see significant effects of genotype, environment, and a genotype by environment effect. And so you can see this here, right? We have genotype 1 on average is growing better than genotype 13 across all different sites, but you still have instances where you see this heterogeneity of variance, right? Where the change in trait value is different across sites for the genotype. So like it's increasing more across sites for genotype 1 than for genotype 13. And you still also get instances of re-ranking where you have, yes, genotype 1 is very good on average, except when you take it to Marilyn Scholl's, where then all of a sudden genotype 13 outperforms genotype 1. The other thing I wanted to point out or mention is that similar to Ford's findings, when you just look at linear extension, so that just the branch lengths that you can take using a ruler or caliper, we actually don't find genotype by environment effects. So this is actually consistent with previous findings. We only see an effective genotype for linear growth, but when you consider this change in higher order growth traits, that's when you start to see these genotype by environment effects. Yeah, and that's just to show you. There's also variation in the capacity for plasticity among genotypes. And what I mean by this is that genotype 1 is sort of accessing a much bigger trait space in terms of how it's changing its morphology across sites than genotype 13, right? Genotype 13 is only changing a little bit. It's plastic, but it's just not going very far across all the sites, whereas genotype 1 is just making these huge morphological adjustments, comparatively speaking, depending on the sites that it's going to. And, you know, we kind of wanted to know, like, does this capacity for plasticity have any benefit? Like, is it good in terms of survival if you're able to kind of shift your morphology? really aggressively across different sites to kind of match what would be the environmental pressures at any given site. And so we ran these correlations. Note, none of these are significant. But what we calculated here was the extent in plasticity. So basically like how much change you saw on average for any given genotype for each of our morphological traits. So this is showing total linear extension, surface area, volume, or the interstitial space. And we related it to the mortality risk. So on the y-axis, this is a Cox proportional hazards risk score, where higher scores is actually a higher mortality risk on average. And so we do actually see negative relationships for all of the traits, where the more plastic you are, the lower your score tends to be, which means that it seems like the capacity for plasticity could be adaptive because it's correlating with these increased survival odds. But we need to dig into the data a little more here because this is really the plasticity of survivors. So this is at a single time point, and we actually have interim data time points where we went and took photographs every 3 months. And we think it's probably going to be more appropriate to look at trait values prior to death. So to say, okay, let's consider the individuals that died. What did they look like before their death time point, and ask how that might correlate with subsequent deaths. So basically like your morphology at the time point prior to death, is there still a correlation there? But I do find this interesting, suggesting that being plastic might be adaptive. So just to summarize this growth data set so far, our initial analysis, we do find genotype by environment effects for both growth and survival. However, we only see a genotype effect for linear extension, similar to what Ford had reported in his 2017 study. Significant interaction effects for all of the other traits, for both growth and survival. And we also find genetic variation in the capacity for plasticity that might correlate with your mortality risk across sites. And what does this all mean? Well, having these G by E effects and the potential for genotype re-ranking, it reduces our ability to predict restoration outcomes. But it also reduces the efficiency of selection if you wanted to say like breed a supercoral, because you don't necessarily have a good handle on the breeding value of any given coral, right? The idea is that you want to be able to say something about the traits that are going to appear in your coral. And if they're totally environmentally flexible, then it's hard to make those predictive decisions. But if you look at aquaculture studies, actually like genotype by environment effects for survival and for other changes, other traits, it's actually not all that uncommon. So this is something that I think we'll have to consider moving forward if we want to try to get into these predictive breeding scenarios. So what could be driving these changes? We see these changes across sites. What environmental variables might be influencing changes in morphology and survival? And again, we're still kind of in the early stage of this analysis. So this is showing some correlation, a correlation matrix for changes in trait values. So volume of the interstitial space, the coral volume, the surface area, and the TLE. So changes in mean trait values compared to changes in some of the water quality parameters, both the mean as well as the range. So we don't have, again, we don't have super high resolution environmental data. So I think these are kind of the best descriptors of the environment that we have so far, sort of like what is your mean, we'll say, nutrient levels. And these are just indicating, sorry, the B indicates benthic. So these nutrient values were taken kind of at the level of the corals for, right, nitrite, phosphorus. We have temperature values, total organic nitrogen. And then we have I think this is silicon dioxide, and light attenuation. So the light attenuation coefficients across the sites. And looking at these, we can see that there's pretty consistent relationships across traits, and it seems like growth is actually positively correlated with nutrient levels and negatively correlated with temperature, which is perhaps not all that surprising, right? You would expect coral growth to be related to temperature. And so we're still working through this analysis and trying to figure out if there's better ways to kind of model the environmental variation. But biologically, the nutrient relationship could have something to do with like fertilization of the symbionts. So having those nutrients that are supporting symbiont communities, and that might be beneficial for growth of the whole animal. And then of course, temperature stress being a negative effect on growth across sites. For the biotic environment, we just actually got our last batch of sequencing data back for our post-transplant libraries on Monday. So I don't have that data to share with you yet. What I'm showing you right now is results from the pre-transplantation study. So for the microbiome, we took samples of all of the corals before we transplanted them to any environment. And then we took samples 1 year later for any surviving corals once they had been transplanted to different reef sites. And so right now, what I have data for is initial differences among genotypes. So all of these genotypes were in a common garden environment, right? So they were all in that nursery environment. And this is showing principal coordinates analysis of the microbial community as measured through amplicon sequencing of the 16S gene. So you can see the corals are different from the water. So these dots down here are just replicates of the nursery water, which is perhaps not surprising, but we have to show that. And then you can actually see a couple clusters in the data where we have, there's a group of genotypes, 3, 7, and 36 that cluster separately from the other nursery genotypes. And when Emily dug into it, she actually found out that it was the abundance of a particular group, so this Rickettsialis, that is kind of driving this difference. So this is now converting that multidimensional trait data into just a bar plot of the abundance of various microbial genera. And this is showing that for these individuals, they actually have significantly less of this, which color is not showing up well, this kind of teal colored MD355, which is not terribly informatively named, but it's actually a type of Rickettsialis. And this is something that I guess has been discovered fairly recently for these corals and seems to be a just something of a consistent signature of Floridian acroporids, is that they can have high abundance of this, what's a newly named species of Aquarium roeri that is an intracellular, potentially an intracellular parasite. And so it seems like our genotypes vary in whether or not they have a very serious like rickettsia infection, we'll say, or not. Some other differences. So this is just another way of showing this, not just who is it, but the abundance of these particular groups. So MD355 substantially lower in genotypes 3, 36, and 7 in comparison to our other genotypes. There was also differences in some other cyanobacteria, actually. So Prochlorococcus and Synechococcus were actually different between, again, in some cases, some different genets where we have different values. So again, kind of take these environmental data with a grain of salt because they're still very much in progress. but it looks like growth does correlate with nutrient levels, but it's anti-correlated with temperature, right? And these actually might be proxies of other overarching oceanographic features like differences in current patterns or upwelling. But I think we'd need to incorporate some different data to kind of test that hypothesis. But it's possible that these environmental variables could be contributing to genotype re-ranking across sites, especially if the responses to those conditions don't necessarily share a common genetic basis. Something else that we still need to fully consider is the potential role of the algal symbionts. So it might not necessarily be host genotype so much as the symbiont genotype that matters. Now for Acroporids cervicornis in the Keys, they're actually all dominated by a single species of symbiont. So it's not as diverse as the symbiosis in some other coral species, but prior microsatellite-based analysis have shown that there is actually strain level variation among some of these genotypes. And so we would have to go back and re-verify this for the fragments that we used in our experiment. But genotypes 1 and 50 were actually some of the most plastic of our genotypes. And it looks like they might host a different strain from some of the other genotypes that we used in our study. So there's still some more work to be done there. And in terms of the microbiome, we also had starting differences in the microbial communities among host genets. So prior studies have suggested that Rickettsia are generally parasitic. There's been work showing associations with coral disease, and new work by Becky Vega-Thurber's group suggests that this particular species that tends to infect Acroporids cervicornis might reduce coral health by consuming nutrients and energy, which would weaken and then eventually kill the host cells. But that being said, we don't actually find a significant relationship between Rickettsia abundance and survival. So those genets that had the lowest Rickettsia abundances, 3, 7, and 36, do tend to fall in the top half of performers. But there's also genotypes that had like reasonably high levels of Rickettsia that are also top performers. So it's not like a silver bullet here. And another question that we still have yet to answer is just how stable is the microbiome? So does that proportion of Rickettsia change, or is it modified in like a site-specific manner? And so the T1 data are hopefully going to help clear up that question and allow us to integrate everything together. So obviously, finishing up these actual analyses is our priority number one and sort of a where are we going with this kind of study. But I'm really excited by the results that we've gotten so far, because I think it just supports asking so many other questions. I'd love to know, what's driving this plasticity? Like, how is the coral sensing its environment and changing its morphology? Like, where does that trigger come in? For corals that are really changing their phenotypes across sites, right? If you're growing up in these different reef environments, is any of that heritable, or does it sort of get reset in every generation? And we're lucky here in that these transplants are still living and ideally will stay living. And they should be reproductive as early as 2022. And so we could actually go out and start to collect spawn from the same genotypes that have now been growing at different sites to kind of ask about some of these transgenerational effects. I'd also love to know if being plastic comes with a cost, right? If you're a coral that's spending all of this energy changing your morphology, do you have to pay the piper at some other time in your life? Like, does it potentially relate to your capacity to withstand temperature stress when it happens? Or is it just a freebie and you can do all of these things, and there are some individuals that can just perform well? I'd also love to start thinking about the bigger ecosystem implications of this capacity for plasticity and these interactive effects, right? Because we're working in this system where we have an ecosystem engineer. And so we would be able to ask, like, what does the ultimate ecosystem look like if you're using corals that are really flexible in their morphology versus others that are more fixed in the morphological space that they can access? And does that change the ultimate reef that you can create through restoration? And we haven't even started looking at the Palmata yet, so it'll probably be a large number of years before I can actually answer all of these questions. But I'd love to hear your thoughts and feedback on this, as well as on some of the analyses we've been doing. And so with that, I will thank my team and my funding agencies again and ask you all if you have any questions.
That was so great. Thank you, Carly. I think,
Yeah,
like the importance of your work, like you said, in terms of informing some of these more progressive things that are being considered in terms of like selectively breeding thermally tolerant corals or super corals, and then actually understanding whether or not those traits hold up when you outplant them into a variety of different environmental conditions is just so critical for actually understanding restoration plans. So I think I just really love your work and what it tries to answer. So right now, if you have questions, oh, I see. Okay. We just have a comment so far. Wonderful presentation. If you have questions, go ahead and just type a question mark or your question directly into the chat box, and we can unmute yourself and then just ask Carly directly. Shannon.
Hi. Okay, so thank you. That was really, really fun. So comprehensive. All the questions you're trying to knit all together into this system. So I'm going to ask a microbiology question. I'm really intrigued by the intracellular rickettsial infection bacterium, and I wonder if, I know you looked at genetic interactions with the host coral, but what about genetic interactions with the dinoflagellate strain, since they're both intracellular? Is that a crazy question?
No, I think that's an interesting question. And something that, yeah, since we haven't verified the strain level information for the symbionts yet. But yeah, that's definitely something that would be worth considering. I mean, at this point, it doesn't seem like there's a systematic difference just based on the strain level identities or the information on the strain level identities that we do have. So knowing, for example, that it's like 1 in 50 that differ based on the symbiont strains, whereas for the Rickettsialis, it was that 3-7,
right.
3-7 and 31. But that's not to say that there couldn't be other variables that we haven't yet considered. So yeah, what I'd really love to do is kind of integrate all of these different lines of information, maybe in some sort of bigger network analysis to try to get a more holistic picture.
Thanks, it's really cool.
Morgan, do you want to go ahead and unmute yourself?
Hello. I was just curious if you could talk a little bit more about how you analyze the microbiome data.
Emily, I will say, has analyzed it like every which way from Sunday. Like Emily's never satisfied, we'll say, like in a good way. And she really wants to be convinced that the pattern that she's seeing is real. And so, yeah, she's literally done every single analysis under the sun, like normalize the data, not normalize the data, run it through DE-seq, run it through principal coordinates analysis. And really we're seeing this same signature. So no matter what we do, we pretty much see the separation where we have those 3 genotypes that have that Rickettsialis signature compared to the others. And it's always the Rickettsialis and those cyanobacteria that pop out as being the main drivers. So we're pretty confident that that is a signal that we're seeing in our data set.
Great, thank you.
You mentioned that Rickettsialis is common in Floridian acroporids. Do you know if it's common throughout the Caribbean?
So I had a conversation with Steve Vollmer about this, and it sounds like Florida might be weird. And so I'm not a microbial ecologist by training. So any of the other microbial people, please feel free to jump in here. But it seems like some of the Florida lines might be different in just the predominance of that Rickettsialis in those particular lines, but I don't actually have great data for other locations. This is just based on a conversation with Steve, who has been working at some of the other Caribbean locations. And I don't think he necessarily sees that huge signature of the rickettsialis like we do see for some of these Florida lines.
Interesting. Ale has a question.
I'm just curious to know, Carly, if you guys remove the rickettsialis, if you think, I mean, it'd be good to remove them and see if you get any other pattern. And as you were saying before, like trying to separate what's related to the genotype and what's just to as a way to see that change. And perhaps, I don't know if you, I guess you guys also planning to follow up on time and see how that community, if it's stabilized in a particular stage or if it changes.
Yes, so if you get rid of the rickettsialis, there's not such a huge difference in the genotypes. And I was just scrolling through my supplementary slides, and unfortunately I don't have that picture, but Emily definitely did that. You lose some of those big genotype specific differences if you remove the rickettsialis. Like I think the cyanobacteria are still showing up, but it's not like the rickettsialis is definitely that main driver. For over time measures, so we sampled them at the 0 point and at the 1 year time point. We didn't repeatedly sample them in the middle, so I don't have a total temporal change of what's going on. So we just have that 1 year, 1 year at the end. And the rationale for that was that we were trying to control for seasonal variation. So we did it at a year in case there were seasonal patterns. Like we really wanted to try to just have it as a genotype and site for that point in time. But you're right that there could be other interesting things going on.
Darrell.
Hello, Carly. Can you hear me?
Yes.
That was a great talk. And people have been touching on the microbe side of things. So I thought I'd just ask more about the evolutionary ecology. And I have two things. One is a sort of general question and a comment. So first of all, yours is the best example of the sort of idea of nested G by E interactions being important in evolutionary ecology that I've heard in the last couple of years. So that's awesome.
Thank you.
And secondly, has there been much advances in the theory of how to analyze nested G by E now that we have so much great genomic data and we have great ability to characterize the environment? And finally, the comment is, does it include structural equation or directional modeling in any way?
So I don't have a great answer for that yet. I mean, it's a great question, is sort of how to integrate all of this information together. And I did just have a conversation with sort of a network analyst earlier this week, in fact, to try to think about, are there kind of non-traditional ways to think about integrating all of this information? It sounds like network analysis might be an interesting thing to think about. So sort of basically trying to do big like correlational networks and then asking how the different hubs and edge weights change. So asking not so much about like individual to individual effects, but like the overall structure of a particular network. So if you think about like, you know, when we actually have the genetic data, you know, if you see a change in the genetic network, is it a single gene that's driving it versus like thinking about, is it the whole genetic network that's changing? And that is the signature of the change in morphology, or is related to the change in morphology as you move across the different sites in time. But I need to dig more into it because again, I'm also not I'm sort of a jack of all trades, master of none when it comes to my science. So I'm hoping to get some more insight. I've played with structural equation models before. And I think that that's actually another good suggestion for thinking about how to integrate across levels. So I'm going to write that down. And thank you.
Yeah, it's very cool. Can't wait to see more. Thank you.
Shannon just dropped a link in to the chat box for her office hours / happy hour that happens on Thursday afternoons. So Carly and everyone that's on, if you would like to transition over to that Google Meet link for a virtual drink and to continue conversation, that would be really lovely. Are there any more questions before we let Carly go? All right. Well, I'd just like to thank you again, Carly, for such a wonderful seminar. And thanks everyone for attending. Next week, we have a former Hope for Reefs postdoc, Hoodson, will be giving us a seminar next Thursday. So we'll be happy to welcome him back. So please join us, hop over to that link that Shannon dropped in the chat box for a virtual drink. Carly, you are welcome, but not obligated. And thanks, everyone. Hope you have a great rest of your day. Thank you so much, Carly, for a great talk.
Cool. Thanks, everyone.
Thank you. Yay.
Right. We'll see if I can jump to that link for a bit anyway.
Figure out how to stop recording.